How about now? Good? Good now? Okay. I'm just hopefully not going to take the entire hour and 45 minutes that was slated for me. The unfortunate part about it is that I could easily fill an hour and 45 minutes and probably even an entire week talking about NMR. Unlike most of the other structural methods, I mean, you know, crystallography, cryo-EM, HDX, cross-linking mass spec, there's essentially kind of like one workflow. And with NMR, there's actually many, many different types of ways you can use it in your studies. So hopefully what you'll gain from my talk is maybe why you should use NMR, or why you should, or why should you use NMR would be a question. And I would pose to you why you should use NMR instead. How NMR can be used to extend your work or perhaps uniquely define your system, and some general information about sample requirements, preparation, data collection, and analysis. A little bit less on the analysis side, but mostly what are the main types of experiments you might be able to use, and how they might complement other data that you may have. So the NMR instruments that are at least directly available to the Hive through us at Scripps, Florida, we have a 700 megahertz Bruker spectrometer, and we recently purchased this 600 megahertz Bruker spectrometer through an S10 grant through the NIH, which in part was funded through contributions that are slated to be for the HIVE Center. So this instrument's ready for y'all to use. It's finally installed and ready to go. We had some issues with the cryoprobe that we ordered. They didn't want to deliver it for over a year because Bruker did not anticipate that they were going to get such a massive influx of people wanting to buy stuff. They're basically the only NMR instrument vendor now. So people with older Varian systems, they can't buy Varian cryoprobes or upgrade their Varian consoles anymore. So they can basically still keep their Varian magnet or Oxford magnet, whatever they had. But then now they have to buy a Bruker console and get Bruker cryoprobes. And so basically Bruker's sort of getting double the amount of business, and they jacked up their prices ridiculously. And so most of the use on biological samples are multidimensional NMR types of experiments. So I'm sure everybody in this room has probably used 1D NMR in organic chemistry class. So small molecules often have very few protons, so they're easily separated, and you can analyze that. Complex carbohydrates have many, many more proton signals, and so what we need to do is to separate this out into multiple dimensions, whether that's 2-dimensional NMR, 3-dimensional NMR, there's even 4D, 5D, ND NMR, you can do like 7D NMR. I mean, you're not actually creating whatever the complicated or structure of a 7-dimensional object is, but it's actually just really associating different chemical shifts. So in a proton NMR, this HSQC experiment, there's nitrogen on this axis, proton on this axis. Really what we're doing is we're correlating the amide proton to nitrogen. So this can occur in the backbone amides. In this experiment, you can also pick up side chain arginine NHs. You can pick up lysine NHs, but these rapidly exchange with solvent, so they're often not visible. There's carbon versions of this, but then in 3D experiments, we're just correlating more nuclei together. So it could be 2 proton dimensions and a nitrogen dimension. It could be proton, carbon, nitrogen dimension. Really what we're trying to do is separate our peaks out, because we're going from something that looks like a big giant mess here that looks to, a little bit more easily analyzable, but still, when you get into this region right here, there's a little bit more overlap. You can get that overlap separate out into 3 dimensions. So NMR is complementary to other structural methods. So some pros versus cons. NMR can provide atomic level insight. It's good for studying disordered proteins, transient interactions, timescale dynamics, and allostery. And it's good, and what we actually like to use it for also is to validate that your structural findings obtained in solid state, such as a crystal structure, extend to the solution state. Because I'm sure everybody's familiar that crystal structures are really valuable, but they can be influenced by crystal packing interactions, or the fact that you're capturing essentially a low energy conformation that may not be fully representative of what your molecule is doing in solution. Cons, it can certainly be size limited. So anything smaller than about 20-30 kilodaltons can be difficult. You can alleviate this by some labeling methods, which I'm going to discuss briefly. Probably amongst all the techniques that you're going to use, it probably requires the highest sample concentrations. Usually when I'm working with a new collaboration, they're like, well, what are your sample concentrations? So I'll give them the volume first. I'll say, it's nice if we can have somewhere between 350, 500 microliters. and then I'll say, at a minimum, we want to target probably like, if we're just doing 2D NMR, you could probably get away with 50 to 100 micromolar. But with NMR, really, it's like the more you can give us, the better, because we can really increase our signal to noise, drop the amount of time it takes to collect data, and really depending on the spectral properties of your sample, if you're working on ubiquitin, which is a really small protein, you can get to 2 millimolar. All of its peaks are really nicely well defined. You could go down to probably 10 micromolar and still actually get really good data on that. But for a lot of interesting things that we're all working on, these proteins have a lot of dynamics, and that can be fast, slow, and what we call intermediate on the NMR timescale. And the intermediate timescale dynamics actually becomes a problem in that the peaks disappear. But sometimes you can actually see these peaks sort of pop up, and so that in these regions where they have these kind of intermediate timescale dynamics often map to functionally important regions. So those are actually regions that we want to observe. So again, NMR can be size limited. So this is an example of ubiquitin. This is an example of maltose binding protein. This is actually a very poor example for me to show to you of a large protein. It's actually a very nice looking NMR spectrum. This and a couple of other large proteins were studied by Louis Kay's lab in Toronto. And actually he's gone through quite a bit of effort to try and find like really nice looking large protein NMR samples so that he could develop the methodology that we can now all apply to study large proteins. But the caveat is maltose binding protein is an interesting protein. Some of the other ones, but a lot of the stuff that we're going to study, they're never going to look this good. But you can see here, as you go to a larger proteins, you get more peaks, you get more signals. You have to keep track of these. You have to correlate them somehow. And in addition to being able to resolve them, just connecting them and trying to figure out, like on a residue by residue basis, which peak belongs to which residue becomes a pretty big challenge. So high sample concentration. There's really not much to get around this. We just require reasonably high sample concentrations. We can use lower volume sample tubes. So this is a normal tube that you might have used in organic chemistry class. There's these tubes called Shigemi tubes, which basically instead of this, you would fill to about 550 microliters. You can put about 325, 350 microliters in this. And the way that this thing works actually is that there's these little NMR, something like that, but basically you can sort of shrink the area in the NMR tube where your sample resides, and when you put your NMR sample into the NMR magnet and you're applying these radio frequency pulses, because these little plugs are sort of tuned to the same frequency that your water sample is tuned to, it allows you to sort of use a lower volume. If you were to just try and take this tube right here, put a lower volume into it, and then sort of center it where the coil should be, you'll get very poor water suppression and probably very broad peaks, and your data would not look good. So, and the requirement for high sample concentration can be offset partially through the use of cryoprobes. These are cryogenically cooled probes. This is really a standard thing these days. So it's not even something that people say like, oh, I'm using cryoprobes, so I get better. It's sort of a standard that people are going to be using this. The way the cryoprobe works essentially is that it cryogenically cools the electronics in the probe essentially. So it doesn't actually increase your signal, it just reduces the noise. But that effectively results in an increase in signal to noise. And so in terms of cons, NMR can be used to determine three-dimensional solution structures, but they can be difficult, and there's several issues associated with this. As you go larger, there's more and more NMR signals that you have to deal with. Not only, so you need to be able to assign chemical shifts to all the protons and carbons and nitrogens in the backbone, but then down the side chain. And then floppy regions will not be as well defined, but they really aren't either in X-ray crystallography. And I don't think that cryo-EM has really gotten to the point where they can deal. I don't even know if they. Basically flexibility is interesting and important, but being able to capture those flexible things is a different matter. And this is actually an example of one of the projects that I did as a graduate student that we determined this actually was a beast of a protein. Actually, I studied two really difficult proteins as a graduate student. First of all, I solved the NMR solution structure of a 30 kilodalton protein, which at the time was like the highest resolution NMR solution structure, one of the highest, I should say. That probably took me about 2 years to do, just analyzing the NMR data. This actually was really interesting but difficult. It was a repeat protein. And there's a repeat region that interacted with each other. So you're looking for sort of like NOE connectivities between this region, but they have very similar chemical shift values. It just became a really big pain in the butt. But what we know by dynamics analysis is that this loop region right here is actually really floppy. And I think a lot of people, when they download PDBs from the PDB files from the data bank, and they just display this information, they look at that and they go, oh, that's floppy, that's flexible. Well, with NMR, actually, this doesn't really kind of tell you about the conformational heterogeneity that this loop undergoes. Really, this is just the limitation of the restraints that we use to determine the solution structure. So you can perform NMR relaxation analyses and say this loop is dynamic, but basically I could take this really well-defined protein structure and remove 90% of the restraints and it's going to look like crap. So really it's like if you have crap in, you get crap out, but the better detailed information that you put in, the higher precision of structure that you'll get. So how do you get started? So first of all, you really want to determine an appropriate strategy. So the best thing to do if you're not familiar with NMR is, you can contact me to discuss. Is your system NMR visible? Meaning, when you make proteins in E. coli without introducing any isotopic labeling, the protons by default are NMR visible, but nitrogen and carbon, the natural abundant isotopes C12 and N14 are not NMR visible. C13 has a very small natural abundance, but it's not enough to really do anything with. So we have to make isotopically labeled protein. So we would do either N15 and or C13 uniform labeling. So you get your entire protein labeled with this down the side chain and the backbone. If you're working in a large protein, we might have to deuterate the protein. So this actually helps in some of the 3D experiments that you might run, helps increase the signal to noise because it doesn't allow basically the signal to sort of dissipate down to the side chain. And to do this, you would grow your E. coli cells in at least 70% D2O, if not nearly 100% D2O. So they don't like it, they will do it. You will often, as you start to introduce these isotopes, you sort of lose more and more protein that you're able to produce. Sometimes you're lucky and you don't. But there are protocols where you can basically do like a step-by-step introduction of deuterium that sort of like initially challenges them, but sort of gets them used to being in a deuterated environment. And then we can also use fluorine. Fluorine's nice because, yes.
When you say large, how large are you talking about?
Large proteins, or so there are NMR studies up to a megadalton. Again, this is like Lewis K's lab where they have very specific things that produce very nicely defined data. I wouldn't say that if you had a megadalton system or even like a 200 kilodalton system that we could do NMR off the bat. NMR is one of those techniques where if we have an idea, we have to make a preliminary sample, collect the data, and say, does this look feasible? Can we tweak it to make the data look better? But certainly you can go to hundreds of kilodaltons using specialized techniques. You're not going to be doing uniform labeling and getting chemical shift assignments for the entire backbone and side chain. We'd probably be doing things like methyltrosy, where we'd be looking at methyl groups. But often those can be the most important probes you might need to look at to tell you something about your system. Fluorine is nice because it's not really biologically present in nature. And you can introduce fluorine, for example, through covalent attachment of essentially like a small molecule that sort of looks like beta-mercaptoethanol, but it has instead of a methyl group, it has a trifluoromethyl group. And it's actually almost as sensitive as protons. So after we make your NMR labeled sample, the first step is preliminary data. How good does the initial 1D or 2D NMR spectrum look? If it looks bad, we'll need to do some optimization. You can modify your construct. Maybe if you had a floppy region that you weren't sure was floppy and it sort of looks like based off the preliminary 2D NMR data that you do have a floppy region and we want to cut that off, we can basically design new constructs. Changing buffers can often help, but if your data look good, there's a number of things we can do. So if your sample looks good, what's next? Obviously, one thing we want to know is which NMR peaks essentially belong to which atom or residue, et cetera. So for some cases, there may be chemical shift assignments already available for your sample. These may either be already deposited into a database that's available online. Before that database existed, people threw stuff into supplemental data. So that's a little bit trickier to go in and find. But sometimes we're lucky and we at least have a starting point. If not, we'll need to prepare, if we have a good sample, probably a double labeled protein, C13 and 15 labeled protein, and perform 3D NMR experiments so that we can assign it. There's also ways that we don't have to do that if you have very specific questions that I'll sort of get into here in a second in terms of specific labeling. But even if we do have a system where your data looks good and there are chemical shift assignments available, we'd still probably want to do some of this work to make sure that what was deposited is actually correct. Because there have been cases, like we worked on a sample, for example, where if you think of a 2D NMR spectrum and you have a bunch of peaks here, well, they must have, you can basically define the chemical shift range where you collect your data, and they may have cut it off at a certain range, but there were actually additional peaks, and they didn't have those when they made the assignment. So probably about 10% of the assignments that they did make were incorrect. And then also, obviously, they didn't assign the things that they didn't see. So what can you do without assignments? Even if you just had just a general sample, does something bind? So you might, it's a little bit tricky with 1D NMR, but you can basically take two interacting molecules in RNA and protein, for example, actually would be good for NMR, because RNA or DNA, they have like regions of the proton spectrum that are populated where proteins are not. And you could just determine, is something binding? Is my protein or sample disordered? Actually, NMR is a really, really good way to figure that out. What you might find for a well-folded protein, you get like a large spectral dispersion of peaks. And for a disordered protein, because things are essentially in a random coil environment and all very similar chemical environments, you get a very narrow dispersion of peaks. And especially there's a region where you see side chain glutamine and asparagine side chains. Instead of getting like this nice kind of well-folded envelope, you essentially see two sharp peaks. And the same thing with methyl groups. All the methyl groups end up being the same chemical environments. You just see one big broad peak in the methyl region. This is actually this is an example. We have a collaborator who was working on a lipid binding protein. They were trying to delipidate the protein. And so they had this complicated procedure that involved a detergent, and then they tried to get crystals of their apoprotein that was delipidated, and they just kept getting these kind of like crystalline looking objects that were very kind of like spongy, and they couldn't get actually something that diffracted. So they sent us their sample that they had prepared, and it turned out that their detergent essentially, I don't know if it encapsulated their protein or just co-purified with their protein, but their detergent actually was still there in super, super high abundance. So even after size exclusion chromatography, even after dialysis, the majority of their sample was this detergent. And it was very obvious by NMR. Like when they sent us the sample, they're like, okay, you're gonna tell us we have a dynamic protein, it doesn't want to crystallize. And I'm like, okay, what's actually in your buffer? Because they sent it to us in a buffer that should have been essentially NMR silent. It was like phosphate buffer and salt. But there were these giant detergent peaks, so it became really obvious. And I just basically did, he said, oh, we use this detergent CTAB, and I Googled, CTAB NMR spectrum, and that's what popped up. It was exactly it. So NMR can actually be used as a detective, essentially, to, if you have an issue of your sample, if it's not behaving the way you think it should behave, it can actually be used to sort of like determine potentially what's wrong. Yes.
Did you determine how much?
Maybe. And actually, if you zoom down, like I'm just showing here, there's so much detergent in the sample that it essentially wipes out all the aliphatic. So this is the aliphatic proton region right here. This would be the backbone amide region, the methyl groups over here. There's so much detergent in it that the proton signals, or the protein signals, barely observable unless you like really, really increase the contour level. It's hard to say if you could do that or not, because I don't know the specifics of the system. I don't know if it's encapsulated or if it's just co-localized, but maybe they're the same molecular size that whatever the micelle of the detergent was, because even a small molecule, I guess if it was spinning at the same rotational correlation time, that it might correlate. It's hard. I would say it's hard to say. I haven't done the experiment, but I wouldn't. Sometimes with some buffers, definitely if you have two different components, like a protein and an RNA, you might be able to determine that. With detergents, I'm not sure though. So uniform versus specific versus segmental labeling. So these are all different types of labeling strategies you could apply to a system. So uniform, I think, is the most commonly used, the one that people maybe know the most about. You use a minimal media, E. coli growth. Protons are naturally abundant and NMR active, so we have to incorporate C13 using C13 labeled glucose, which is about, I can't remember exactly, but it's probably about $150 a gram. For a liter, you probably need about 3 to 6 grams of this glucose. So, you can do the math. I can add up quickly. N15 labeling is actually pretty cheap. It's only about 10 to 20 bucks a gram. In deuterium, we have to grow the bacteria in D2O, about 70 to 100%. And I'm trying to think how much, that might be on the order of about $550 a liter or so. And supposedly you can retain the D2O that's been used and send it back to the company, and you might get a credit for for giving them something that they can then purify and resell. Essentially, they wouldn't sell. I don't know if they can make 100% D2O after that. If it's maybe that goes into that 70. They have like some weird stock. It's like 77.3% D2O or something. I don't know. Yes. Yeah. And we hide it. Actually, a lot of organic chemistry labs I think have D2O, but they just use very small vials. And we have giant liter bottles. We're not like mega stocked out, but yeah. So yeah, they basically sell them in big jugs, just like you would buy PBS or something like that. But definitely they're more expensive. So it's also possible to label in yeast, in insect cells, and mammalian cells. We haven't done this in part because it's super expensive compared to E. coli, and we really haven't had a need to do that, but it's definitely possible. So you can also label specific amino acids. So if you had even a small or large protein, it doesn't really matter, but maybe you're only interested in like a particular acid, amino acid type, or maybe there's even like a very specific residue that that you're interested in. There's ways that we can sort of isolate that. So you could, like for example, only label valine residues with N15. And this is actually a really tricky one where you perform so this is a 2D HSQC. This is a 2D HSQC, but it's essentially a 3D version where it's sort of like running an HNCO experiment where it's trying to correlate an amide proton to nitrogen to carbonyl. But what they've done is that they know they had a valine with a leucine -1. So they just C13 labeled leucine, N15 labeled valine, everything else was unlabeled, and only that peak, well, actually there were a couple of peaks here. You can kind of see here, like this was the N15 valine in black, and in blue are just the ones that had the leucine -1. So there's ways that you can even isolate just more specific residues if you wanted to, just by choosing how your labeling scheme. And then you can also label specific nuclei, like methyl groups. So this is what you would use for larger proteins. You can specifically label methyl groups, and actually it can be very complicated. Methyl groups have 3 protons. You can just get 1 proton labeled, 2 deuterons down the side chain. This is actually what you would use for very large systems, because deuterium allows you to collect highly sensitive data on large proteins. This is an example where I'm trying to remember. I think that this is uniform labeling, and then here they did like this very specific methyl labeling. So this would be just like if you make C13 N15 labeled protein. This is a large protein. I can't remember how large it is. Actually, this might have been the Megadalton. I think this is the Megadalton paper. You don't really see any of the methyl peaks. But when you use this methyl labeling protocol, where it's very specific for large proteins, you can actually get these peaks to show up. The problem that I've been thinking, I'm not quite sure how they do this, but basically, how do you identify which peak belongs to which residue in such a system like that? Because you can't run all the sophisticated 3D NMR experiments to do that. One way that you could do is mutagenesis. Yep, for sure. So then there's also segmental labeling. So you might have a protein that has 2 domains, and you want to know maybe what the influence of one domain is on the other, because a lot of times what we're doing is we're crystallizing one domain, not considering the other one. So you can use either this sortase-based approach or like an N-tene system-based approach. Actually, it's vice versa. This is sortase, this is N-tene system. And essentially what that does is, you could collect, for example, here in red black is an NMR spectrum of a 2-domain protein, and in red is an NMR spectrum of where they've segmentally labeled one domain and the other domain is unlabeled. And that's sort of a little bit more visible here, where you can see there's a bunch of black dots. Here there's 3 red dots, and they overlay with a couple black dots. So you could isolate one domain within the context of a larger protein. So this can actually be interesting too if you're studying like a multi-domain protein that has intrinsically disordered regions. You could specifically study the intrinsically disordered region when it's bound to the rest of its structured protein, for example, to determine, if you remove the disordered region from the rest of the protein, does it have a structural change, or is it the same in solution? Things like that. And then you can use 19F labeling on your protein or molecule, again, by introducing cysteine. So this is an example of the PPAR gamma ligand binding domain that Pat told you about a little bit earlier. This actually had a cysteine present in wild type that we had to mutate out so that we could introduce one here where we wanted to have it. And this actually is a very efficient reaction. You can get almost 100% labeling with this. To do NMR, sophisticated NMR studies, we need to do NMR chemical shift assignment. That typically involves 3D NMR data collection. Again, there's also 4D, 5D, and ND methods where you can basically correlate a bunch of information together. Once you get beyond 4D, my brain sort of explodes. I'm still not sure how they handle all that data, but those methods are out there. And typically what you're doing is you're taking this like 2D HSQC experiment, you're sort of just expanding those peaks out into multiple dimensions into this third dimension. So usually it's in these assignment experiments, it's a carbonyl dimension or a C alpha dimension or a C beta dimension. And you can step through the data just by kind of in the program by basically kind of mousing up and down, and you can see sort of peaks appear and disappear, appear and disappear. It's a little bit shown schematically here, where you're basically just taking this 2D NMR spectrum and you're kind of stretching things out into 3 dimensions. And so what we'll do is we'll collect 3D NMR data to kind of make these correlations. This is a backbone amide. In this experiment, the CBCACONH correlates this backbone amide to the -1 C alpha and C beta. And then there's a complementary experiment called the CBCANH that gives you the same correlations, but it also gives you the intra-residue correlations. And so what you'd see if you went to a specific, let's see here, like you chose like this peak right here, and you went to that region in the 3D NMR data set, and you align those planes, what you would see for the CBCANH, you'd see 4 peaks, 2 C alpha peaks, 2 C beta peaks. For the CBCACONH plane, you'd see only 2, and there should be a correlation where you see basically, and actually usually in this experiment right here, the -1 peaks are usually lower amplitude, and the I peaks are usually higher amplitude, so that's another way you can determine those. But basically what you're doing is you're kind of collecting these types of 3D data. There's experiments to collect carbonyl data too, and you're kind of comparing these information to known average chemical shifts of C alphas, C betas, carbonyls, and all different types of residues. So alanines, for example, have very unique C beta chemical shifts. Serine and threonines have very unique C beta chemical shifts. They show up down here in the 60s. And then the other ones have some differences, but they're a little bit more trickier to kind of differentiate. And so actually, oftentimes if you're working on a larger protein, in addition to these types of data, you might even collect data all the way down the side chain to give you the gamma, the delta, the epsilon, things like that. And those actually really tell you what the -1 residue is, because like arginines and lysines have very unique chemical shifts profiles. And so you link all these things up basically using a computer software, or like when I was a graduate student and didn't really have sophisticated computer software, you could use a Microsoft Excel spreadsheet. That was a pain in the butt. But basically what you're trying to do is you're trying to link all these different spin systems together. So again, each one of these kind of strip plots belongs to one of those peaks in the HSQC spectrum. And what you're doing is looking for things that correlate together. So these guys correlate this way, and then there's another peak that correlates to this, and there's 2 other peaks, and that correlates to that. So you can kind of link all these things together. When you get to proline, you stop because prolines don't have backbone amide groups, but then you can kind of continue on from there. So ways that you can use NMR in your biomolecular studies. So I'm not really going over any of the theory or kind of like how to collect the data, how to analyze the data, but I wanted to kind of give you guys some ideas of the general types of NMR experiments that you might be able to use in your studies. And then if you have questions, maybe we can talk later or something like that about how NMR you might be able to apply it to your system. So if your protein is small, you can't do cryo. If it's floppy or otherwise doesn't crystallize, you could try to solve an NMR solution structure. And so the basic way this works is that we basically do a computer simulation where we have your protein primary sequence that sort of like is in an extended disordered kind of computational state, and we feed it a bunch of structural restraints to fold it up. So for that, we'd have to perform additional 3D NMR data collection to assign the side chains, because that becomes important. We collect and analyze NMR data to get structural restraints, in particular these nosy data, which gives you information about how close protons are. And then we use these in an iterative structure calculation, essentially kind of like over and over again, where we do a calculation, go back to our data, we kind of say, and there's ways to sort of like ambiguously assign chemical shifts and NOEs, but as you kind of go through these iterative processes, it becomes more and more apparent what the real answer is rather than the ambiguous statement. So nosy experiments provide NOE restraints. These are localized sort of phenomenon that provide information on short, medium, and long range distances. So they are distance dependent, 1 to the -R6. They detect spins that are about 5 angstroms apart, so protons that are about 5 angstroms apart or less. And the intensity of the NOE cross peak is related to the distance between these 2 different protons. So if you have a very intense one, it means they're closer. If you have a very weak one, it means they're farther apart. And you can calibrate these intensities into distances, or you can group them into kind of distance ranges. strong, medium, and weak. So this actually, I was afraid to pull up. I wanted to show an example of like, this is the backbone amide. These are all the different protons that are nearby. I actually pulled this up from my dissertation from I don't even know when that was, but I vowed to myself I would never open that PDF ever again, and I did it for you all today, actually. So these are actually strips of a 3D NMR experiment. These are strips from a 4D NMR experiment. The nice thing about, so 4D NMR, it's not as sensitive, but the nice thing about it is here, these strips down a 3D experiment, the peak could belong to several different types of protons. Through iterative assignment, we figured out what they belong to, but when you separate this out into 4 dimensions, even though the signal to noise is not as good, what's nice is that you can get the separation not only on the proton dimension, but then also either on a nitrogen dimension or a carbon dimension, and then it really becomes obvious which peak or which residue these peaks essentially belong to. You get torsion angle restraints. In some cases, you can measure these directly using specific NMR experiments. But the most common method is that once you have your NMR chemical shift assignments for your backbone nuclei, especially the alpha and beta carbons, but also carbonyl, nitrogens, and amide protons can be predictive. If you compare this information to a database of structured proteins that's been developed, and then their chemical shifts, if you compare those chemical shifts for things that are either an alpha helix or a beta strand to the random coil values, the information essentially can be predictive of whether or not this amino acid is in a helix or a sheet. And you can kind of see here in purple here are the random coil values. And for the C alpha, you can see that if you have something where the C alpha chemical shift is more positive than random coil, it's in a helix. If it's more negative than random coil, it's in a sheet, and it's vice versa for the C beta. So essentially you can feed distance restraints, you can restrain the secondary structure, and then also you can get information on global restraints using this methodology called residual dipolar couplings. Again, each one of these little things essentially could easily be a very long review article, if not an entire book. But essentially, these types of restraints don't tell you which things are interacting or the secondary structure, but on a global basis, if you think of your backbone amides as like a vector, it sort of tells you which direction those vectors should be pointing. So the NOEs will sort of fold your protein, the torsion angles will put it into a secondary structure, but if you throw that into an NMR structure calculation, you might get fuzzy structures. You might get an ensemble that has it's not very precise. But when you put these residual dipolar couplings into there, your precision will increase. And often what people will do is they'll collect several different types of residual dipolar couplings. You can get a backbone amide, you can get H alpha, C alpha, there's all different types. And they collect 5, they might use 4, and they'll reserve 1 as sort of like a structural validation tool. They'll do the structure calculation, and then what you can do is you can take those structures, you can back calculate what these residual dipolar couplings should be, and then you can compare them to the actual values. And if you're getting good structural convergence, you should essentially get like a linear plot that's like, R equal to 0.99 something, R squared. So you can also do biomolecular complexes. There's ways to get structural restraints between proteins and nucleic acids or 2 different proteins, for example. You can do selective nosy experiments where you might have a C13N15 labeled protein and unlabeled RNA, for example, and you could get information about that. There's also a method called paramagnetic resonance enhancement, PRE, that can be used, it's especially useful for complexes. So this takes advantage of a physical phenomenon that a nucleus with a lone electron can affect the physical properties essentially of another nearby NMR active nucleus. And really what this does is, like, if you think of your HSQC fingerprint spectrum, you introduce a PRE label, anything within a certain distance of that PRE label essentially will disappear because of this PRE effect. And there's a nice distance dependence where if you have very short distances, the peak will completely disappear. And that's, I think, within 13 to 20. I think I didn't copy some of this in here, but there's a distance sweet spot where things either disappear, but then there's a certain distance range where the peak doesn't disappear, but its amplitude will vary. And actually that can be very predictive in terms of like the distance between the PRE spin label and that certain nucleus. So this is actually a very sensitive way to monitor binding interactions, but it's also nice for getting information about weak or transient interactions. So if you have like some interaction that's occurring on the very weak micromolar level or even millimolar, PRE NMR can be very sensitive. I mean, a lot of those times those things are crystallizable. And actually, this becomes kind of very important in this whole liquid-liquid phase separation thing, where you get these like weak binding interactions, but within the context of the phase separation, they can be tighter because the local environment is more concentrated. But then also, proteins move between different conformations, and you can actually see conformations that maybe aren't crystallized that occur in these PRE data. And I'm going to show you a couple examples here. But just like the fluorine label, you would covalently attach one of these PRE spin labels, that are the same labels that are used in EPR, to a cysteine or a lysine residue, collect 2D NMR data in a paramagnetic and a diamagnetic state. So for some of these PRE labels, they can be reduced from paramagnetic to diamagnetic state using like a reducing agent, like beta-mercaptoethanol, things like that. And you're basically just comparing peak intensity ratios, so it's actually a pretty easy way to analyze the data. So this was an example I found in the literature of a protein RNA complex. They're actually doing NMR of the RNA, but then they spin labeled the protein. And it's hard to see here, but essentially they sort of highlighted a couple of RNA peaks that disappeared in the paramagnetic state that reappear in the diamagnetic state. So it's a way that you can sort of site specifically determine where the interactions are occurring. This is something I just threw up actually recently because Pat was talking about PPAR gamma. We've been studying PPAR gamma bound in collaboration with Pat, bound to agonists and coactivator peptides. So that's like the fully active state. And so this right here is helix 12. The ligand binds here. This is the peptide binding surface. I'm not showing it here, but basically we put a PRE NMR spin label here. And what we see is that we saw basically peak intensities either completely disappear or drop on this entire surface, like you would expect from the crystal structure. But what actually becomes really interesting, if you just have the ligand bound without the peptide, or the peptide bound without the ligand, so it's still an agonist without or a coactivator, you don't see the same profile. Actually, helix 12 is dynamic still in those states, and actually adopts a completely different conformation. And we've done this with a repressive inactive conformation too. So you could use NMR chemical shift perturbation to map binding epitopes. So there's so many different ways you could do this: protein ligand, protein nucleic acid, protein metal, nucleic acid ligand, nucleic acid metal. I mean, the sky is sort of the limit. And there's sort of three general phenomena that can occur in these types of data. For weak interactions, you would typically see things that occur in fast exchange. So as you titrate in more and more of your interacting partner, you'll see basically a movement of the peak from one state to another, from the free to the bound state. If you have a very high affinity complex, you'll see something that occurs in slow exchange. So as you start to titrate in the molecule, you'll get one peak disappear while the other state appears. And it's because at 50% addition, what happens is that 50% of the molecules are bound and 50% of the molecules are still free. Whereas with the weak binding one, there's such a rapid exchange on and on and on and off. What occurs is that you're essentially seeing sort of like this population average of your binding. And then there's also an exchange regime called intermediate exchange. It sort of occurs for interactions that occur. So when I say weak, I say it's probably high micromolar, millimolar. Strong binding is like nanomolar binding, even better. And intermediate exchange typically occurs somewhere in the, I'd say, low micromolar regime. So it's actually where a lot of interesting biology occurs, unfortunately. And what happens is that your peaks disappear. So this can actually be informative if you have a protein and you add your molecule, if it's a protein or a ligand or a RNA, it becomes really easy to map where the binding interface is because the peaks disappear. If you know what residues those belong to, you can map the binding epitope. You can also observe, really complicated things too. So you can observe these multi-site binding effects. So, sometimes there's two interaction sites, and what you might see is something ridiculous like this, where you get a chemical shift change followed by some other chemical shift change that moves in an opposite direction. You can also see interesting things like conformational change after ligand binding. So this could be really cool. So you might have like a free state and a bound state here. And if you did a titration, what you might expect to see is that as this peak disappears, it's moving toward the bound state. But oftentimes, what you'll see is that the peak moves in a different direction as this peak appears. And what's happening is that it's binding to the protein undergoes a conformational change that allows maybe the ligand to bind. And so you don't get this direct one-to-one correlation. So these are kind of like the very specific kind of physical phenomena that you can kind of pick out with NMR spectroscopy. So you can determine binding epitopes. So this is a project that we did on the glucocorticoid receptor, the DNA binding domain. So our collaborator had identified a non-coding RNA stem loop that GR was predicted to bind to. So we had a lot of really nice binding data, a lot of really nice functional data through collaboration, but he could not get a crystal structure of the complex. So what we did was we used NMR essentially to map the binding epitopes on both sides. So this is a homonuclear NMR experiment looking at the RNA. We did this with and without unlabeled protein. And so this is actually a region of the RNA spectrum where protein signals don't show up. So we were able to determine which base pairs were essentially affected by the interaction. We did the same thing on the protein side. We basically did 2D HSQC analysis to say which protein residues were affected in the interaction. And then we could actually use that NMR data to essentially drive the docking of these two molecules. And so our collaborator had previously solved the crystal structure of the DNA binding domain isolated. He solved the crystal structure of this RNA stem loop isolated. It turned out that the major groove of the RNA stem loop in the isolated form was a little bit more compressed than what would be probably occur in a complex. So we used molecular dynamic simulations essentially to let it breathe. And I sort of picked out something from the simulation where it was kind of like open enough that we were able then to dock in the protein to the RNA. And then using that RNA model, he identified some base pairs that looked like they would be important in the interaction, mutated them, and they were important in the interaction. So mutation of this base pairs essentially in a fluorescence polarization binding assay essentially sort of killed the binding affinity. So this is a way where you can sort of use NMR to sort of determine, even if we're not doing a high resolution structure, you can sort of get information about the binding epitopes, and you can use that NMR data to sort of drive modeling of the complex. Same collaborator, we studied, we can use NMR to study allostery too. So, crystal structures typically capture a low energy conformation, but proteins can move between various low energy conformations, some of which can be higher energy than the form that wants to crystallize. And so this is a project where our collaborator had identified essentially an ancestral mutation that essentially allowed glucocorticoid receptor the ability to bind to a specific type of response element. That if you basically resurrected different versions of the DNA binding domain, and early on it couldn't bind to it, but there was a mutation that occurred, this glycine 425S, that allowed it to bind to this DNA response element. And so what we did essentially was very simple 2D NMR analysis of what happens when we analyze wild type current glucocorticoid receptor versus that ancestral glucocorticoid receptor. And then we look for NMR chemical shift perturbations, because you can imagine if you're just making this one little point mutant, you might not actually see a dramatic change in your crystal structure. And actually it became pretty interesting. So, right around the site of mutation, obviously you would expect to see NMR chemical shift perturbations because you're changing the local environment around there. But then we also saw some distant allosteric effects. And even though this serine is located essentially on not the nucleic acid interaction side, mutation of this residue, that occurred through evolution actually affected the conformation of residues and regions that interacted with the RNA. So this is a way that we could actually use NMR sort of like to probe allostery. So, distant mutations can allosterically affect functional surfaces, and you may not be able to pick that up with crystallography. So, obviously one of the really good ways of using NMR on biomolecular structural studies is with intrinsically disordered proteins. They're not amenable to crystallography typically unless you have some sort of complex where maybe you get some sort of disorder to order transition where you can get it to crystallize. And you can't study disordered proteins easily or maybe at all with cryo-EM. So NMR is ideally suited to study IDPs. You can study dynamical properties using relaxation methods. You can study these folding upon binding types of phenomena, including NMR structure calculations. And you can study their interactions. So you can study interactions of IDPs with structured proteins and also with other unstructured proteins using chemical shift perturbations and these PRE methods. And you can also, as I mentioned, use segmental isotope labeling. So this was a study that I found, whereas a protein in its calcium-bound form has a very nice dispersed NMR spectrum. In the apo form, however, you see essentially all the NMR peaks collapse into the center of the spectrum. So this is like a classic looking intrinsically disordered protein or region spectrum. And then when they added a partial amount of calcium, maybe 50%, what you see is that, some of the calcium-bound peaks appear, but what you can also see is that there's still a lot of disorder here. So it's probably like 50-50. So this is a way where you can study, essentially disordered proteins and transitions from disordered to ordered. We've used NMR to study, there's a region of transcriptional co-regulators that interact with nuclear receptors, and they have essentially these LXXL motifs that typically crystallize in the nuclear receptor functional surface. But these co-regulators typically have multiple LXXL motifs, and they all have different affinities. And so one question we had is that, if you took a nuclear receptor complex, are they interacting with all three motifs, or is the whole thing interacting sort of like as a global cohesive unit? And so when we first collected, if you scroll back here, these peaks all kind of collapse, but you can identify the peaks. With our protein, though, this looked like crap, essentially. And then, up here, this is the glycine region, the glycine signature region. Our protein, I think maybe we expected to have, 15 resident peaks in this region, and we saw 3. And the reason for this was that more than likely what's occurring is that the backbone amides have rapid exchange with solvent, and they disappeared. But using our cryoprobe, we were able to collect carbon-detected NMR data. So here, this is basically the same nitrogen dimension in each one of these spectra. But instead of proton detected, this is carbonyl detected, this is C alpha detected. And what you can see here is that we're able to separate this information out and actually see essentially all the peaks that we would expect to see. So you can see here there's like 2 or 3 peaks in this kind of glycine region, whereas there's a lot of peaks here. And actually the orangish red is the apo form, and in blue is the form where we complexed it with our protein. But there's different types of these carbon detect experiments. This is one that correlates carbonyl carbons to the nitrogens. This correlates C alphas to the nitrogens. So there's different ways for separating out this information. And what's unique about this too is if you have a protein that's proline rich, high proline content, you can actually observe prolines in these 2D data, whereas you don't observe them in a normal HSQC because they lack amide groups. This was a really cool study that was recently published. This is basically the interaction between 2 intrinsically disordered proteins. So you're thinking 2 intrinsically disordered proteins are really floppy, they're not going to interact with high affinity. This is actually, I think it says picomolar affinity, maybe even it's sub-picomolar, but these 2 intrinsically disordered proteins interact with picomolar affinity. And the reason it's basically driven by charge. One of the proteins is highly positively charged, one of them is highly negatively charged. And it's really hard to see here, but essentially the basically red and blue spectra that are overlaid here, they look really similar because it still retains its disorder, but even though it's disordered, it interacts with high affinity. So, these NMR methods are really one of the unique ways where you can use NMR to study disordered proteins. And this is an example of not only does it not interact with a structured protein, it's interacting with another disordered protein, but you're not getting a disordered to ordered transition. It's still retaining its disorder. So, you're probably not going to be able to crystallize this or study it by cryo-EM. Obviously, one of the very popular ways of using NMR is to measure and study timescale dynamics. So, proteins move between all sorts of different conformations. They can have very fast motions where you're getting rotations essentially of bond vibration, methyl group rotations, side chain rotamers. But then you can also get these more higher energy conformations that can occur on the millisecond, microsecond timescale or even longer, where you're getting essentially larger domain motions. And NMR is very sensitive to all types of motions, from the very fast picosecond, nanosecond timescales up to second timescales. I'm just going to show you a couple of different things that I was able to throw together real quick from some studies that we've recently published. This is a nuclear receptor ligand binding domain, and I won't go through the whole story about what the paper was about, but basically we performed this. It's called the heteronuclear NOE experiment. What it tells you is a little bit about disorder on the picosecond, nanosecond timescale. And in particular, things that have very low heteronuclear NOE values are essentially very floppy on the picosecond, nanosecond timescale. So you might think of them as things that you wouldn't see crystallize. They would either be the ends of the N-terminus or the C-terminus of your tails of your protein are very flexible loops. And in this protein in particular, some people had made some kind of like, oh, there's a structural region and it looks like it might have a unique conformation compared to everything else. But if you look at the NMR data, it shows actually that these things are really floppy and disordered. So it's, basically one of our conclusions was you should probably limit what you're interpreting about the structural region. But this can be a very useful way to study disorder in your protein. Especially if you're unsure, even just looking at crystal structures, you might have certain loops. And, again, if you're downloading a PDB and not looking at the B factors, some of these things are kind of like correlating with B factors, but it's a very nice way essentially to look for disorder in proteins. I'm going to flip through a couple of slides where we are able to use NMR to look at slow exchange between 2 conformations. So this is slow exchange on the seconds time scale. So we were studying these 2 molecules. These are synthetic ligands targeting this nuclear receptor PPAR gamma. We were interested in this in part because The difference between these two molecules really is sort of like a one atom change. You're changing a phenyl group to a pyridyl group. But just that small change essentially turned a neutral ligand into an inverse agonist, into a co-repressor selective ligand. So we saw the crystal structure of one of these molecules and compared it to the crystal structure of the other molecule. And when you overlay the crystal structures, there's essentially no information there about what is the mechanism. Why is one repressing transcription and the other one neutral? This is in part because crystal packing influences the conformation of this critical functional interface. So basically, there's this critical switch helix 12 that Pat's talked about and that I mentioned earlier. There's another helix 12 essentially that comes and binds and stabilizes it into one conformation. We did end up getting some useful information from the crystal structures though, because it turns out that there's a water-mediated hydrogen bond network between this pyridyl group and the arginine that turns out to be important. If you mutate this to an alanine or leucine, essentially this molecule looks dead, just like the covalent antagonist. But structurally, we had no idea what was going on with the conformation of the protein because the crystal structures all look the same. But when we collected 2D NMR data, a little bit hard to see here, but basically in the green spectrum, essentially we had a certain number of peaks. And in the black spectrum, which was bound to this molecule that had the pyridyl group that had the unique functional profile, we saw peak doubling for a bunch of different peaks. And so peak doubling, usually on, if you're an NMR person, you're like, oh, I've got slow exchange between two conformations. And it turns out if you overlay, if you zoom into each one of those peaks, there's a state A and a state B. There's a shared state and a unique state essentially. And this happens all throughout the entire NMR spectrum. So, and we could use these NMR relaxation methods to actually verify that these two peaks correspond to slowly exchanging populations, because you could also imagine you could have double the amount of peaks because, for example, maybe your protein got proteolytically cleaved and half of it is missing part of it. And so that causes two unique chemical environments. But using NMR methods, we can actually observe this exchange. So what occurs is that you've got these two peaks right here. And as you increase this NMR delay, essentially you allow the exchange to occur between those two conformations, and you get these two cross peaks that show up here. And you can kind of fit these data to get rates. That could be interesting if you're studying a kinase where you're wanting to determine if the rate of exchange between two conformation correlates to like an enzyme turnover event or something like that. But what became a really interesting way to use NMR is to look to see how these two populations bind to a coactivator peptide or a co-repressor peptide. So this state, again, was the state that was shared with this antagonist. This was the state that's uniquely populated by the repressive molecule. And it turns out when you add the co-repressor peptide into it, this state right here essentially it's mimicking the co-repressor bound state. So it's sort of pre-populating the state. So this ligand pre-populates something that it wants to interact with the co-repressor peptide with high affinity. Vice versa, if you had the coactivator peptide, it's that other state that pre-populates sort of like an active state. So this molecule, basically its unique function is that it allows the protein to switch back and forth between two conformations, which we're not capturing by crystallography, but we're seeing it by NMR. And with NMR, we can do biochemical titrations and determine what do those two populations like to do, essentially. Does one like to interaction partner or the other? So we can also do ligand-detected NMR experiments. You can directly observe the bound conformation of a ligand. You can determine, does the ligand bind? What is the ligand stoichiometry? And you can do this through a variety of methods, such as fluorine NMR, if you have a fluorine-labeled ligand, essentially, or proton NMR methods, such as STD NMR or water logs. And these will just really, and these methods are good for weakly binding ligands. So these can be used for screens. So you want to screen a molecule of fragments or of weak binding ligands. These are good studies, but if you have a high affinity ligand and you want to validate that it binds to your protein, these are not good approaches to use. You typically need to be in the high micromolar-ish range or better. Or I think you could probably even do some sort of competition experiment where if you did have a high affinity ligand, there's ways you can do that. This is a way that we use fluorine NMR to map stoichiometry. So this is a PPAR gamma agonist, has a trifluoromethyl group. A crystal structure was reported of it where it bound in, basically you see one molecule bound in the orthosteric pocket here. But when we performed a titration of the molecule, we saw one peak appear, and then above stoichiometry of 1, we saw a second peak appear. So basically we're capturing a second binding event that was not captured in the crystal structure. And we use 2D NMR methods. So this is a 2D N15 HSQC experiment. This is looking at the methyl region. We can basically map the binding epitope of that second binding event. So if we have the molecule bound, the protein bound to one molecule, compare an NMR spectrum of that to a second added molecule, what turns out is that In the apo form, we get a transition in slow exchange to that first binding event because this molecule binds with like 2 nanomolar affinity to the orthosteric pocket, but that second binding event turns out to be 2 micromolar instead of 2 nanomolar. So you go from slow exchange, where we're seeing this sort of this peak move, one peak disappears, the other one appears, to essentially right here, this is the second binding event. We can sort of see the peak broaden and disappear. And so we were able to basically map the binding epitope of that second binding event by using this differential NMR analysis. This was a pretty, at least I thought, a pretty cool way to use fluorine NMR to validate crystallized ligand orientations. So this is another type of PPAR gamma agonist that in a crystal structure where there's 2 molecules in the asymmetric unit, there were 3 different ligand conformations observed for this molecule. So the first one here in the orthosteric pocket, this orange group kind of adopts this kind of U-shaped conformation shown here. And the second one, instead of adopting a U-shaped conformation, it adopts a more extended conformation, and then a second molecule was found to crystallize to this other surface right here. So you can kind of see all these different conformations here. And so we used fluorine NMR to say, is this actually occurring in solution? Is this a crystal artifact? And it turns out it is occurring in solution. So what happens is that when you titrate this molecule that has a trifluoromethyl group up to 1 equivalent, you see the population of 2 peaks. And then when you go up to 2 equivalents, those 2 peaks sort of stay there, but you get this third peak popping up. And so what happens is that in this conformation, it's physically blocking that second molecule from binding there. But in this flipped conformation right here, that second molecule can bind. And we validated this with 2D NMR. Basically, if you have a simple binding event, you should see a transition either in slow exchange or fast exchange or an immediate exchange between state A to state B. But what we saw is state A to state B to state C, to a different state. So the NMR data essentially validated that crystal structure. So it's not an artifact. And then you can also use protein-detected fluorine NMR data, again, so you can site-specifically incorporate cysteine. So this can become complicated. If you have multiple cysteines, you'd have to mutate them all out and site-specifically introduce the one you in. There may be ways of using like these, kind of bioorthogonal ways of putting like a natural amino acids. I haven't tried to do that, but if anybody's interested in doing that, that's certainly some way we can, a way we can try to incorporate fluorine. But essentially what we did here was label with fluorine this a residue here on helix 12 that doesn't interact with the coactivator peptide bound at the surface here. And then we just profiled what does the fluorine spectra of this protein look like bound to a bunch of different ligands. So high affinity agonists, partial agonists, non-agonists, repressive molecules. And what we were able to do is make correlations between chemical shift values that we observed, essentially going from, there must be some sort of here. Okay, whatever. Chemical shift values to peptide recruitment efficacy. So we saw these linear correlations between chemical shift or the structural state and the ability to recruit the peptide. Yes.
This work as well with
single fluorine. Yeah, you can do them with multiple fluorines. Like Kurt Butrick's done this with GPCRs, so you can have multiple fluorines introduced. With our system, we don't have to assign the peak because there's only 1 fluorine peak. With that, you'd want to basically prepare a couple of different samples. Let's say if you had 3 cysteines and you wanted to label those fluorines, then you'd have to do like iterative site-directed mutagenesis to identify which peak belongs to which one. You'd also want to be careful from the sense that you want to make sure that the fluorine signals don't overlap. The nice thing about fluorine is that it really has a very large spectral dispersion, so that's typically not a big issue. And we use this molecule called BTFA, and there's a couple of other different ones where basically just changing the chemical environment around that fluorine can really change its spectral properties. Fluorine is super sensitive to environment, but there are ways of basically labeling with multiple fluorines. Actually, this study, which was recently published in Nature Communications, was the first postdoc in my lab who now has his own independent lab. But in addition to labeling with, he had that helix 12 labeled, so he labeled the protein with fluorine. One of the molecules that we were studying also had fluorine and had a trifluoromethyl group. So you could study simultaneously the protein and the ligand at the same time. And it turns out that this fluorine, if we're looking in the range of like 80 ppm or something like that, this is on the protein. The ligand fluorine chemical shift values were like at 60. So I mean, they were way far away, so you could physically separate them in the spectra and still see them. But we could essentially take this information and sort of do like a spectral deconvolution and kind of group these things into different clusters that had different functional features essentially. So Anyway, that's it. 58 minutes. Might be time for happy hour. So if you guys have questions, I'd be happy to take them. If you want to talk later today or tomorrow about ways you might be able to use NMR in your research, I'm more than happy to